All right, so today in biology we need to go over our section 4 notes from chapter 1. Section 4 is about biologists, tools, and technology. So your objectives are going to be to describe the usefulness of modern imaging techniques and to explain the usefulness of computer models in studying biological systems. And also, you need to be able to summarize how modern computer-based technologies have advanced the study of genetics. So we're going to go ahead and get started here. Our most important technology in biology is probably that microscope. All right, so that's what we'll talk about first. Remember that all living things are made of cells, and cells are really, really tiny. We might need to be able to see those cells to understand how they work. So a microscope was a huge advancement in biology. It was a huge technology to further the advancements of biology. And a microscope is an instrument that provides an enlarged image of an object. Right? You guys already knew that. You've probably used a microscope before. But we're going to talk about some different kinds of microscopes that biologists might use, because the microscope that you may have used before probably isn't good enough to be able to see those tiny little structures inside of a cell. Right? You're familiar with a light microscope, and biologists in a lot of cases use electron microscopes so that they can see tiny, tiny things. All right. So this first question here would be a whiteboard question normally, and I'd have you use your book to figure it out, but since it's actually kind of complicated, I might just give it to you this time. All right, so it says, how do scanning electron microscopes and transmission electron microscopes produce different images of the same specimen? So you're going to need to understand how those two types of microscopes are different and how they're similar. Right? They both use electrons. Right? They're bouncing electrons that are tiny, tiny off of these particles, off of these cells, off of these specimens, whatever it is that you're trying to take an image of. And electrons, right, will bounce off of things much, much smaller than light will bounce off of. So you get much higher resolution, higher magnifications of whatever it is that you need to be looking at. So they both use electrons to get an image. But one of them sends the electrons through the image, and then you try and figure out what was there by what's missing. And the other one actually bounces the electrons off of the surface of that specimen, so you need to understand the difference. Right? When you're doing the scanning electron microscope, right, that's actually just going to bounce electrons off the surface of your specimen. And so what they would do actually is take this cell or whatever it is that you're looking at, and they coat it with a very thin layer of metal and then bounce electrons off of it to see what that surface looks like. Right? It just lets you know what those external features of the cell are, how it looks, what it's shaped like. Right? And those are going to be three-dimensional images at the end of the day. Right? Bouncing those electrons off the surface, you should be able to get a 3D look of what that surface looks like. Okay, and the transmission electron microscope, that was a little bit different. Right? It's going to take a tiny sliver of that specimen, so you actually have to cut it into cross sections first, and then you shoot your electrons through that specimen and it's kind of like an x-ray. You know how they send that radiation through your arm, your leg, whatever it is that's being x-rayed, and really you get a negative image. You see the bones on that x-ray because the radiation actually couldn't go through in that spot. So you kind of get a reverse image. But when you're sending the electrons through the specimen, you should be able to see those tiny, tiny little structures inside of that cell. It's going to give you a two-dimensional picture though, but that two-dimensional picture is going to have a much higher magnification. You can see things that are a lot smaller than in the case of that three-dimensional SEM picture. Right, so one of them bounces electrons off the surface, you end up with the 3D picture. The other one sends the electrons through the specimen, and you end up with a 2D picture. But one key also that you should throw in with that transmission electron microscope is you're probably going to end up with a much higher magnification as well, because you can see things that are smaller and smaller in that technique. Okay, on this next slide, we have 3 more questions, right? 2 of them having to do with microscopes, and you can answer those on page 21 in your book. And then the third one's talking about computer models, and I believe you can answer that on page 22 in your book. Yes, just check that out. So it is on page 22. But these are things that you're going to answer after we're done with our notes. And then you're going to have a quiz online that checks to make sure that you found the answers to these things. So make sure that you're doing that appropriately. You want to have good answers, but we'll do that in a minute. Okay, so some other definitions from this section that we need to know. First off is a gene, right? And that's going to be a segment of DNA containing genetic information. You've probably talked about genes before. You've talked about your genome. You've talked about genetics, right? All of those have the same root word. And basically, it's that genetic information. It's all those things that are you, right? So you have a specific gene that makes your eyes whatever color your eyes are, whether they're blue, green, brown, whatever. You have a specific gene that leads to specific proteins that are inside of your cells working as enzymes, or maybe they're structural proteins, whatever they are. Right? So basically everything about you comes from your genes. Right? It's part of your DNA. Remember, that's what's inherited. That's what you pass on to the next generation. That's what you got from the generation before you. Molecular genetics is the study and manipulation of DNA on a molecular level. So we're going to get into that in depth when we get into our genetics unit in biology, which won't be until a little bit before Christmas, I think. But we'll get into that a lot, talking about how that DNA, how that molecule works, and how it, right, what we know about how it works, and what that means as far as how are you. Genomics is a study and comparison of genomes both within and across species. So when you look at those collective genes that are inside of each of your cells, and then you compare those to other humans, and then maybe even compare those to other animals in general, right? Genomics talks a lot about how different things are related, right? If you see similarities in DNA, those things might be related ultimately because they would have inherited that DNA from a common ancestor. So we'll talk about that a lot later as well, right? Not just during our genetics section, but also in our evolution unit when we start talking about how all these living things might be related to each other. Now, these things specifically, as they apply to this section, right? A lot of what we know about genetics, a lot of what we can do with genomics is completely dependent on computers, right? We've gone through the process of actually mapping out the entire human genome. And it took 10 years using computers to do that. So it's really, really complicated. There's lots of variables. There's lots of things to look at any given time. So it's really, really important that we have the technology that we do. This is the part of biology that's completely different than what your parents learned when they were in high school, right? Because we didn't know this stuff at that time. So this is relatively new to biology when we're talking about genetics, genomics. So that's going to be pretty important for the future of biology. So that's why we're focusing on it so much. Okay, and then we have this last prompt here for your whiteboard. Right? Again, we don't really have to use a whiteboard today because I'm not here with you. But I would like you in your notes to provide an example of how technology has helped biologists gain a better understanding of life. And we talked about a technology right at the beginning of these notes that was pretty dang important to help gain a better understanding of life. But any example will work. And remember that technology is just something that anything that's useful and helps with your day-to-day life. It doesn't have to be high-tech. It doesn't have to be a computer. It doesn't have to be your cell phone, right? It can be extremely low-tech as well, right? The wheel is a piece of technology. Now that specific piece of technology is not a very good example for this, but you should be able to come up with an example of how technology has helped biologists gain a better understanding of life. So make sure that ends up in your notes too. Other than that, we're done with this section, right? Right now, you need to go back through and answer some questions using your book, and then you're going to go get a computer and take a quiz over those questions. So make sure you do them appropriately. You will be graded. Good luck.